Cancer cells are notorious for their relentless growth and proliferation. To sustain this aggressive behavior, they require a constant supply of energy and building blocks. This is where metabolism comes into play. Metabolism refers to the complex network of chemical reactions that occur within cells to convert nutrients into energy and essential molecules. In the 1920s, German biochemist Otto Warburg made a groundbreaking observation. He noticed that cancer cells exhibit a peculiar metabolic characteristic. They prefer to produce energy through a process called glycolysis, even in the presence of ample oxygen. This phenomenon, known as the Warburg effect, has puzzled scientists for decades. Normal cells typically rely on a more efficient process called oxidative phosphorylation, which takes place in the mitochondria, the cell's powerhouses. However, cancer cells seem to favor the less efficient glycolytic pathway, shunting glucose away from the mitochondria and producing large amounts of lactate as a byproduct. This metabolic reprogramming gives cancer cells a significant advantage in the battle against healthy tissues. By altering their metabolism, cancer cells can thrive in harsh environments, resist cell death, and even evade the effects of some cancer treatments. To understand the Warburg effect, it's essential to grasp the basics of normal cell metabolism. Imagine our cells as bustling cities, constantly consuming fuel to power their activities. Glucose, a simple sugar obtained from the food we eat, serves as the primary energy source for our cells. Normal cells employ two main pathways to extract energy from glucose: glycolysis and oxidative phosphorylation. Glycolysis is like a quick energy fix. It occurs in the cytoplasm, the cell's interior, and involves a series of chemical reactions that break down glucose into pyruvate, producing a small amount of ATP, the cell's energy currency. Oxidative phosphorylation, on the other hand, is a more elaborate process that takes place in the mitochondria. Pyruvate produced during glycolysis enters the mitochondria and undergoes a series of reactions, ultimately generating a much larger amount of ATP. This process requires oxygen and is therefore called aerobic respiration. In the presence of sufficient oxygen, normal cells prefer oxidative phosphorylation as it is far more efficient at producing energy. Glycolysis, being less efficient, is generally reserved for situations where oxygen is limited, such as during intense exercise. Cancer cells, however, defy this metabolic logic. Even when oxygen is readily available, they exhibit an insatiable appetite for glucose and predominantly rely on glycolysis for energy production. This metabolic shift, known as the Warburg effect, provides several advantages for cancer cell survival and growth. Firstly, glycolysis produces ATP much faster than oxidative phosphorylation, albeit in smaller quantities. This rapid energy production fuels the accelerated growth and proliferation of cancer cells. Secondly, glycolysis provides cancer cells with building blocks for synthesizing new proteins, lipids, and nucleic acids, essential components for cell division and tumor growth. Thirdly, the increased glycolytic flux in cancer cells leads to the accumulation of lactate, a byproduct of anaerobic metabolism. Lactate creates an acidic environment around the tumor, which can suppress the immune system and promote tumor invasion and metastasis. The Warburg effect is a hallmark of cancer and is observed in a wide range of tumor types. Understanding the underlying mechanisms driving this metabolic switch is crucial for developing new therapeutic strategies to target cancer cells selectively. The Warburg effect, first observed nearly a century ago, has been the subject of intense research and debate. Scientists have made significant strides in unraveling the molecular intricacies of this metabolic adaptation. Dr. Thomas McFate and his colleagues at Duke University made a groundbreaking discovery that shed light on the role of lactate in cancer progression. They found that high lactate levels, a hallmark of the Warburg effect, can actually protect cancer cells from destruction by the immune system. Lactate accomplishes this by suppressing the activity of T cells, a type of immune cell responsible for recognizing and eliminating cancer cells. This finding highlighted the importance of targeting lactate production as a potential therapeutic strategy. Another seminal study by Drs. Gopinath Sutendra and Evangelos D. Mikalakis at the University of Alberta provided further insights into the Warburg effect. They discovered that cancer cells often exhibit reduced mitochondrial activity, despite having functional mitochondria. This mitochondrial dysfunction further drives their dependence on glycolysis for energy production. Their research revealed that cancer cells frequently inactivate a key mitochondrial enzyme called pyruvate dehydrogenase, PDH, which is responsible for converting pyruvate into acetyl-CoA, a molecule that enters the oxidative phosphorylation pathway. This inactivation of PDH effectively shuts down the mitochondria's ability to utilize pyruvate, forcing the cell to rely on glycolysis. Section 5. Exploiting the difference, targeting cancer metabolism. The distinct metabolic profile of cancer cells, characterized by their reliance on glycolysis and altered mitochondrial function, presents a unique opportunity for therapeutic intervention. Researchers are actively exploring various strategies to exploit these metabolic vulnerabilities and develop novel cancer treatments. One promising approach is to directly inhibit key enzymes involved in the glycolytic pathway. By blocking these enzymes, researchers aim to starve cancer cells of their primary energy source and halt their growth. Several glycolytic inhibitors are currently under investigation in clinical trials, showing potential as anti-cancer agents. Another strategy focuses on reactivating mitochondrial function in cancer cells. By restoring oxidative phosphorylation, researchers hope to shift cancer cells back to a more normal metabolic state, potentially making them more susceptible to conventional therapies like chemotherapy and radiation. Targeting lactate production is another avenue being explored. By inhibiting the enzymes responsible for lactate production or neutralizing the acidic environment created by lactate, researchers aim to enhance the immune system's ability to recognize and destroy cancer cells. Beyond the Warburg effect, the complex landscape of cancer metabolism. While the Warburg effect has long been considered a hallmark of cancer, it's important to recognize that cancer metabolism is a complex and dynamic process. Not all cancer cells exhibit the classic Warburg phenotype, and metabolic adaptations can vary significantly depending on the tumor type, genetic background, and environmental factors. For instance, some cancers exhibit increased activity of certain metabolic pathways, such as glutaminolysis, which utilizes the amino acid glutamine as an alternative fuel source. Others may upregulate lipid metabolism, utilizing fatty acids for energy production and building blocks. Furthermore, recent research has revealed that metabolic interactions between cancer cells and their surrounding environment, known as the tumor microenvironment, play a crucial role in cancer progression. Cancer cells can manipulate neighboring cells, such as fibroblasts and immune cells, to support their metabolic demands. Therefore, a comprehensive understanding of cancer metabolism requires going beyond the Warburg effect and considering the intricate interplay between various metabolic pathways and the tumor microenvironment. The promise of metabolic therapies, a new frontier in cancer treatment. Targeting cancer metabolism holds immense promise for developing more effective and less toxic cancer treatments. Compared to traditional therapies that target rapidly dividing cells indiscriminately, metabolic therapies offer the potential for greater selectivity and fewer side effects. Since cancer cells rely on specific metabolic pathways for survival and growth, targeting these pathways could potentially spare normal cells, reducing the harsh side effects often associated with chemotherapy and radiation. Moreover, metabolic therapies may be effective against drug-resistant cancers that have developed mechanisms to evade conventional treatments. Furthermore, combining metabolic therapies with existing treatments could enhance their efficacy. For example, inhibiting glycolysis in cancer cells may make them more susceptible to chemotherapy drugs that target DNA replication. While metabolic therapies are still in their early stages of development, several promising candidates are showing encouraging results in clinical trials. As our understanding of cancer metabolism deepens, we can expect to see more innovative and targeted therapies emerge in the fight against cancer. Challenges and opportunities. Navigating the road ahead. While the field of cancer metabolism holds tremendous promise, several challenges and opportunities lie ahead. One challenge is the sheer complexity of metabolic networks and the dynamic interplay between cancer cells and their microenvironment. Developing effective metabolic therapies requires a thorough understanding of these intricate interactions and the ability to identify specific metabolic vulnerabilities that can be exploited without causing significant harm to normal cells. Another challenge is the development of accurate and reliable biomarkers to identify patients who are most likely to benefit from specific metabolic therapies. However, Advancements in technologies such as genomics, proteomics, and metabolomics are providing researchers with powerful tools to unravel the complexities of cancer metabolism and develop more precise and personalized treatment strategies. The growing knowledge of metabolic reprogramming in cancer is also opening up new avenues for early detection and prevention. By identifying metabolic changes that occur during the early stages of cancer development, researchers hope to develop strategies for early intervention and potentially prevent cancer altogether. Conclusion: a metabolic revolution in cancer care. The discovery of the Warburg effect revolutionized our understanding of cancer biology and paved the way for a paradigm shift in cancer treatment. Targeting cancer metabolism represents a promising new frontier in the fight against this devastating disease. While significant challenges remain, the ongoing research and development in this field hold immense promise for improving cancer care and transforming the lives of millions worldwide. As we delve deeper into the metabolic intricacies of cancer cells, we move closer to a future where cancer is no longer a death sentence, but a manageable condition. The journey ahead is filled with both challenges and opportunities, but the pursuit of understanding and exploiting cancer metabolism offers a beacon of hope for a brighter and healthier future. The metabolic revolution in cancer care has begun, and its impact is only just beginning to be felt.